Okay. Hi everyone. Thank you so much for coming today, both in person and those online. My name is Betsy Boughton, and I'm the program director of agroecology at Buck Island Ranch. And today we have one of our awesome interns presenting, Emmy Stewart. Emmy came to the ranch in August of 2023, so she's been here about 6 months, and she graduated from University of North Carolina, Chapel Hill in May 2023. And her research, she has different experiences, some working on farms, edible campus, UNC. So she's been interested in agriculture for quite a while. And she's also worked on different ecosystem function projects, like looking at greenhouse gas flux in streams. And she's done various field ecology research assistants, one in the Galapagos. That's super awesome. And worked on birds there. And also has a Spanish minor and is a translator. So that's really cool. So Emmy was a really positive force in our lab, and I really enjoyed having her, and I know that our whole lab did. She's always cheerful, always easygoing, laid back, ready for anything. And she had a really interesting project because she collaborated with our long-term agroecosystem research network folks. So she's sort of got to work in this network science and get exposed to collaborations all the way in Colorado. So she also did a lot with remote sensing in our program, our data analysis, which she'll tell us about. After she leaves here, she's going to Lake Powell in Arizona to be a kayak and a stand-up paddleboard tour guide. And I'm super jealous of that. And then afterwards, she is exploring graduate school and maybe even continuing on with remote sensing and agriculture. So really excited to see where she goes and hope you stay in touch. But today I'll turn it over to Emmy, and she's going to talk about producing remotely sensed measurements of standing biomass at Buck Island Ranch.
Thank you, Betsy. So yeah, my project today is on producing remotely sensed measurements of standing biomass at Buck Island Ranch. So when I say biomass, I'm referring to the above ground biomass, above ground standing biomass in the pastures, both the entire vegetation structure above ground, both the living and the dead vegetation. And this is important because it provides a habitat to a lot of different organisms and is also forage for the cattle on the ranch. Understanding the amount of biomass is important because grazing lands provide a variety of ecosystem services for us. And so in order to sustain these services, we have to know how much biomass is there. And the amount of biomass also informs adaptive management, meaning making decisions based on ground conditions at the time. And so it's important for ranchers to understand the amount of biomass in the different pastures to know when and where to move their cattle and when and where to conduct prescribed burns. We've been collecting biomass at Buck Island Ranch since 2017. It began as part of a National Institute of Food and Agriculture project to look at the effect of patch burning on biomass. The map here shows the original pastures that were used in this project, but currently we collect biomass in around 10 to 12 pastures. Each pasture has 9 points. And to collect the biomass, we have cages set up in the pastures that are 0.25 meter squared areas that exclude the cows from grazing. So we clip that biomass, and that is the non-graze biomass estimate. And then we have a paired plot that we also clip that's the same area, and that is representative of the pasture as a whole. This method of clipping biomass to understand the amount that's there is very time and labor intensive. So we're interested in exploring alternatives to clipping in order to save time and money. The potential alternative to clipping is using remote sensing to estimate biomass. There are a couple of benefits to using remote sensing. We have publicly available data sets that have the ability to estimate large areas of biomass accurately. These estimates also have real time or near real time. However, we do have to have those ground measurements in order to build these models to use remote sensing. The remote sensing data set that we used for this project was the harmonized Landsat and Sentinel data set. Over there, you can see let's see if this works. Okay, here you can see the tiles of this data set. It's based on a modified military grid reference system on a UTM projection. And we get 30-meter resolution every 2 to 3 days. This combination data set is a project between NASA and USGS. And with the Landsat and the Sentinel satellites, we get surface reflectance data and multispectral indices. Here you can see what the images look like when they're first taken from the satellite, and then atmospheric correction is done on it to remove clouds and shadows. So then this is the picture of what it's going to look like when you're deriving the indices from it. This project took place at Buck Island Ranch, which is a subtropical cattle ranch. That's about 10,500 acres. We have about 3,000 head of cattle at any point. And we have both native, semi-native, and improved pastures. The improved pastures consist of introduced grass, primarily bahia grass, that has high palatability and the most biomass available in the summer months. We also have semi-native grass or semi-native pastures that have slightly less palatable species but sustain their biomass year-round. So using these two types of pastures together allows for year-round grazing. So as Betsy mentioned, this was a collaboration with another LTAR site. The Central Plains Experimental Range in Colorado developed this workflow in 2022 to estimate biomass at their site. So Buck Island Ranch, along with a couple other LTAR sites, were all working through this simultaneously to see how or whether this workflow could be used in different regions, and also to hopefully eventually create a web tool with real-time biomass estimates for ranchers to be able to use. So their workflow was to pull the satellite images with the same data set, the HLS data set, extract them and process these images, and then use ground data that they collected to tune and fit the model. And then with that ground data also to validate the model and assess the accuracy of it. They collected their ground data in a slightly different way. They used non-destructive measurements or methods, which allowed them to have more estimations of biomass over a larger area. So some of the satellite vegetation indices that they used were EVI, which is a measure of greenness overall. It's able to calculate dense vegetation accurately, and it doesn't saturate very easily. We also use NDVI, which is similar to EVI, but it does have the tendency to saturate with high levels of greenness, especially in the summer months. They also use the shortwave infrared band, which is less sensitive to the photosynthetic state of plants, but is affected by moisture. Then a couple of the other ones they used were some individual color bands. They used these newer broadband spectral angle indices that some people have had really good luck with. And then there were 28 indices that they used in total. The variables that we selected for our model, well, we had the dependent model as above ground biomass. And then the vegetation indices that we selected were chosen after looking at all 28 indices together and removing the ones that had high correlation. And then we also had a variable for pasture type, so the semi-native versus improved, seasonality, the wet versus dry season, and a random effect to account for the differences between pastures, like elevation differences or spatial location. Our hypothesis for this project was that the EVI indice or the shortwave infrared band would be one of the best predictors of biomass, because the EVI has the ability to calculate dense vegetation, and the SWIR because it's less sensitive to the photosynthetic state, and it also was one of the top variables at CPER in their models. We also hypothesized that pasture type would be an influential predictor of biomass because improved pastures have higher homogeneity. So this is a time series of our first hypothesized indice, or the one that we hypothesized would be one of the better predictors. Here it's the measure of EVI, and then the month, and each by year. So we would expect, because EVI is a measure of greenness, that in the summer months it would be higher, and in the winter months it would be lower. And you can see that that trend does occur here. And we also have a time series for the shortwave infrared band, which we would expect the inverse of, because the shortwave infrared band is measuring the reflectance of shortwave infrared light. In the summer months, when there's more greenness and more moisture, more infrared light is absorbed, so less is reflected. So we see the inverse of what we just saw, how it's lower in the summer and higher in the winter. The methods for this project were to first compile the data from our 6 years of collected ground data at Buck Island Ranch. This data was then sent to Dr. Sean Kearney and Erica Pierce at CPER to extract the satellite tiles and process it. They processed it with 3 different masks to remove clouds and shadows. We then, with the returned indice calculations, assess the multicollinearity between them to build 2 linear mixed effects models. We used AIC for variable selection, and then also did a principal component analysis where the calculated principal components had a very weak correlation with standing biomass. And then with our models, to assess their predictive power, we looked at the correlation between the observed and model predicted biomass, and using R² and the root mean squared error to compare the two. And then we also cross-validated the model spatially and temporally. So these are the correlations between the standing biomass and the model predicted biomass for models 1 and 2. You can see the variables that were put into the models up here. Both of the models have a moderate correlation and a fairly similar root mean squared error. Model 2 has a slightly higher correlation and slightly lower root mean squared error, but they're fairly similar. They both have a considerable amount of noise. So to temporally cross-validate the models, we removed 2021, the randomly selected year 2021 from the data, rebuilt the models on the remaining data, and then predicted, or then reran those models on 2021. So this is the observed standing biomass from 2021, and then the new model that was not trained on 2021, but predicted on 2021. And again, there's a moderate correlation for both models 1 and 2. And this is telling us the ability of the model to predict in new years. So similarly, to do the spatial cross-validation to determine the model's ability to predict on new pastures, we randomly selected a semi-native and improved pasture to remove, rebuild the model on the other pastures, and then predict on pastures J and B. Again, there's a moderate correlation here. We then calculated the relative variable importance for each of the models. This tells us that despite all of these variables being left in, some of them are more important than others. So we calculated the sum of all the AIC weights of all the models that had that variable in it to show that for model 1, seasonality and the blue band were the most important. And in model 2, the shortwave infrared band, the BAI 136 band, and the NCI band were the most important. To revisit our hypotheses, our first hypothesis was that the EVI indice and the shortwave infrared band would be the best predictors of biomass. EVI being a predictor or a good predictor was not supported. EVI was an indice included in Model 1, and it had a low variable importance. And the shortwave infrared band was included in Model 2, and this was supported as a good predictor because it had the highest variable importance in Model 2. We also hypothesized that pasture type would be an influential predictor of biomass. This was not supported as pasture type. The pasture type variable was removed from Model 1 in our variable selection stage, and in Model 2, it was left in but did not have a high variable importance, high relative variable importance. Compare these results to those at the Central Plains experimental range. Their top model here had a high correlation with their observed standing biomass. This could be because their location is a lot more homogeneous. They're in Colorado. They also don't have as much seasonality, so that could be a factor in it. As I mentioned before, they also collect biomass in a different way, so they were able to have more measurements and over a greater area, so that also could have contributed to having a higher correlation. The implications of this are that these model inputs and the biomass that we've collected at Buck Island Ranch don't allow us to build a model with a strong enough correlation to use to predict biomass. So currently, clipping or using non-destructive methods is still the most effective way to measure biomass at Buck Island Ranch. However, there were moderate correlations. So this encourages us to continue to explore the capability of satellite remote sensing to predict biomass, potentially collecting our ground truthing data in a different way, maybe more similar to how they did at CPER, could be a way to do that. It also leads us to want to try to use UAVs or drones, because with the high resolution that those could provide, they could gather data that would allow a higher correlation. I want to thank everybody at CPER for helping me, specifically Dr. Sean Kearney and Erica Pierce, the Agroecology Lab at Buck Island Ranch, specifically Greg and Betsy, and everybody at Archwell. So thank you. Do I need to? I was like, am I allowed to do this?
Okay, I'll have you speak into this. We're going to have questions now.
Hi, Emmy. That was a great talk. I wanted to ask you, what do you think are the sources of the other 60% that you couldn't explain? So your model explained about 40% of the variation, right? And so the 60% of the variation is not explained. And I was wondering, what do you think were the sources of?
That's a good question. I don't really know. It could be that we've selected indices that yeah, I really don't know. Yeah, that's a good question though.
I'll just interject there. I think it could be due to like just having 9 samples in a big pasture, like 9 samples representing a 30 by 30 meter grid. So I just think we're not really representative of the tile that the satellite's measuring, but.
Okay. What's next, Sterling? Come over here, Aaron.
All right, yeah, great talk. I was curious, like, what would like functionally happen with the grasses as they're growing and producing standing biomass? That like, how would that link to the features that you're actually trying to measure? Like, do they start reflecting more or less as they're higher, as the grasses start growing higher up, or like, how does that work with what you would predict?
I think it depends on the indice, but specifically, like, so as the area increases, they'll have more area to reflect back. But they calculate different things. So like for the shortwave infrared, I would assume that's the case, that like the more biomass there is, like the larger it is, then it's gonna be reflecting. Yeah, and I guess also like depending on or not if they're like in fluorescence, and that could affect like the color bands, whether they're being picked up or not. Let me see if you have any online. Let me see what.
Oh, I have a question. So, what do you think is the best scale for your estimate measure? Okay, is it best to estimate like a global scale or like a state scale or range scale, something like that?
Yeah, that's a good question. I feel like some sort of regional scale would work. I think it would depend on like how the ecosystems vary within it, like how diverse it is. But maybe more like ecosystem scale is a better one, because I think like, like if you are doing it in Florida and you have all these grazing lands, but I don't think it would be, it would work. I think that ideally you would be able to use one model to predict everything, but I think it has to be ecosystem specific because of the different plants that are in each area. All right, Greg.
To follow on that, have you tried to kind of combine all your 9 plots per pasture into an average and look at the average Landsat value for that pasture as well, and looking at the relationship between those two to assess a little bit better, like if the scales at which you're looking at is influencing your capacity to predict?
I think that was one of the next steps to look at. This one was by plot scale or like by sample scale, but I think looking at it by pasture scale maybe has an ability to look at those averages there.
I have a question for Akeem. What is in your hand in this photo?
Do you want to take a guess? Actually, a soil sample that looks like dookie.
It does.
Oh,
It really does.
Yeah. It's moist soil being sieved.
Okay,
Yeah.
that's good to know.